Hello there, it's Chloe, and for today's video, I will going to talk about the two stages of photosynthesis, which is the light-independent reaction and light-dependent reaction. But before all of that, I would like to discuss first what is photosynthesis. So photosynthesis is the process which plants use sunlight, water, and carbon dioxide to create energy and oxygen in the form of sugar. So as I mentioned earlier, I'm going to talk all about the two stages of photosynthesis, which is the light-dependent reaction and the light-independent reaction. So first, let us have the light-dependent reaction. So in light-dependent reaction, we have the sunlight here that would be, the light energy that would be absorbed of the thylakoid, and the H2O as well will be absorbed by the thylakoid. So when the water and light energy will occur inside the thylakoid, it would be that will be the time that the oxidation will happen. So the water will oxidize and will turn into oxygen. But always remember that every oxidation, it will always have reduction reaction. So after the oxidation, there will be two electrons that will remain inside the thylakoid. That would be picked up by the NADP plus or nicotinamide adenine dinucleotide phosphate. So after the NADP plus pick up the remaining two electrons here inside the thylakoid, it will turn into NADPH or nicotinamide adenine dinucleotide phosphate hydrogen. And next, so as we remembered, there is also light energy remains inside the thylakoid. So that light energy that remains here inside the thylakoid will be also picked up by the ADP or the adenosine diphosphate and will turn into ATP or adenosine triphosphate. So that is the simple way, or that is the simple explanation of the light-dependent reaction process. But let me tell you as well the reactants of the light-dependent reaction. So the reactants of light-dependent reaction is the ADP, phosphate, NADP plus, and the water. While the product would be the oxygen, ATP, and NADPH. And now let us move to the light independent reaction, or which also known as the Calvin cycle. So this will be occur in stroma. So now in the Calvin cycle or light independent reaction, the NADPH here will oxidize, and as you remember, after every oxidation or reduction reaction, there will two electrons that will remain, and that two electrons will be received by the carbon dioxide. So by the time that NADPH oxidized and two electrons was received by the carbon dioxide, the NADPH will be back again in NADP plus. So after carbon dioxide received the electrons from the oxidation that happened from the NADPH, carbon dioxide needs energy to be able to push through the process so that it can be result as glucose or sugar. So the energy will be given by the ATP. So ATP will give the energy to carbon dioxide, and the ATP again will be back in ADP. So after the energy given by the ATP to carbon dioxide, the carbon dioxide will push through or will have the power to be able to push through the process to become glucose or sugar. So in that case, the process has been done. But now I will going to tell you what is the reactants of the Calvin cycle. So the reactants would be obviously the ATP and NADPH and the carbon dioxide. And the product would be, of course, glucose or sugar and ADP, phosphate, and NADP plus. So that would be all for the simple explanation of these two stages of their different processes. So now let us dig deeper to these two stages of photosynthesis. So my partner will be tackle about the deeper explanation of these two cycles. So may I call on my partner, which is George Cagolata.
So now, I'm George Cagulada. So let's dig deeper in the light-dependent reaction. So as you can see, let's zoom in the thylakoid membrane. So this is the thylakoid membrane. This is the stroma, and this is the lumen or the inner thylakoid space. So in the thylakoid membrane, you can see two photosystems. This is the photosystem 2 and photosystem 1. Photosystem 2 is also called P680 because this is the maximum wavelength that this photosystem can absorb. And photosystem 1 is also called P700 because this is the maximum wavelength that this photosystem can absorb. So let's start with the process. So in the photosystem 2, the electrons in the photosystem 2 will excite when the light will hit these electrons and will go to PQ or the plastoquinone. Since the electrons here are transferred into the plastoquinone, this needs another electrons. So this is when the time that the water or H2O will enter the process. So the H2O will be oxidized and give its electrons to photosystem 2 and will give off one oxygen atom and two hydrogen ions. So let's go back to the electron that excited in the chlorophyll. From the photosystem 2 to plastoquinone, it will go to the cytochrome B6F complex. When these excited electrons will flow or will go to this cytochrome B6F complex, its energy will produce or will be used as a pump to pump hydrogen ions from the stroma into the lumen or the inner thylakoid space. And after that, these electrons will travel to PC or plastocyanine. And from the plastocyanine to photosystem 1. So from here, the electrons, because of the electron transport chain, it loses its energy. And because of that, it needs another energy or it needs light energy to excite itself. So in the photosystem 1, the light will struck the electrons again and will be excited and go to FD or the fluorodoxine. And after that, the electrons will go to NADP reductase. NADP reductase is a protein and it is also a enzyme, as you can see, because it is tase in the last part of whenever there is tase in last, it is automatically be classified as an enzyme. So from here, the electrons will go out from the NADP reductase and will be accepted by the NADP plus along with the hydrogen atoms and reduces itself into NADPH or nicotinamide adenine dinucleotide phosphate hydrogen. And since when the cytochrome pump in hydrogen, it it will lead to high hydrogen concentration in the inner telecode space. So because of that, the hydrogen will need to go to a process which is called chemiosmosis and will flow in the ATP synthase. So ATP synthase is an enzyme used to make it to make ADP into ATP. So when the hydrogen ions will flow through the ATP, this particular protein will rotate and eventually will combine ADP and phosphate and turn into ATP or adenosine triphosphate. So here, that is the process on that is the process in the light dependent reaction. So is I will dig deeper what are the processes that happens inside the Calvin cycle. Calvin cycle can be divided into three parts. First is the fixation of CO2. Second is reduction of PGA to G3P. Third is regeneration of RUBP from G3P. First, so let's start. First, the carbon here will enter the process. And as it enters the process, it will automatically react with ribulose 1:5 biphosphate. And it will be catalyzed by the enzyme which is called rubisco. And because of that, it will result into 3-phosphoglycerate. And that are the things that happened in the fixation of CO2. But before we move on to reduction of PGA to G3P, we will track how many carbons are being used in this process. As you can see here, there is 3 ribulose 1:5 biphosphate, and it contains 5 carbons. So 5 carbons times 3 is automatically equals to 15 carbons, and we have 3 carbons here, so 15 plus 3, it will automatically equals to 18 carbons. And because of that, when ribulose 1:5 biphosphate reacted with the carbon that enters the process, it will initially form a 6 carbons that carries 2 phosphate groups, and this will be divided into 3. So 18 divided by 3 will result into 6 PGA or 6 3-phosphoglycerate. So that is the things that happen in the fixation of CO2. Next is we move on to reduction of PGA to G3P. As you can see here, we have a PGA kinase. PGA kinase is an enzyme that catalyzes phosphate, turning it into a molecule. So in this process, we need 6 ATP since we have 6 PGA. When ATP here enters the process, it will turn into ADP and give it phosphate into the 3-phosphoglycerate. So this phosphate will be received or will be accepted by the 3-phosphoglycerate or the 6 PGA, and it will looks like this. It carries 3 carbon and 2 phosphate groups, and it is called 1:3 biphosphoglycerate. Next, in the next process, we need an ADPH or nicotinamide adenine dinucleotide phosphate hydrogen. And as you can see here also, we have a G3P dehydrogenase. So this enzyme is the one that will reduce or that will reduce the I mean that will turn NADPH into NADP plus. When NADPH enter the process, its hydrogen will be removed together with its phosphate. So phosphate here, one of its phosphate will be removed. And as NADPH enters the process, it gives its 2 electrons to 1:3 by phosphoglycerate in order it to turn into G3P or glyceraldehyde 3-phosphate. And because that its phosphate, one of its phosphate is removed, it will look like this. It carries 3 carbon dioxide, and 1 phosphate group. And it is called glyceraldehyde 3-phosphate. And because we have 6 1:3 by phosphoglycerate, we will also have 6 glyceraldehyde 3-phosphate. As it continue the process, one of the G3P will go down and will be used to produce sugar. So we will move on to the third stage of the Calvin cycle, which is the regeneration of RUBP from G3P. As what I said, one of the five G3P will continue the process, while one will go down and will be used to produce sugar. The 5 G3P here it carries equivalent of 15 carbons. And as what I said earlier in my discussion, a ribulose 1:5 biphosphate or RUBP contains 5 carbon and 2 phosphate. And because we have only 1 phosphate here, we need something that can give us phosphate. So that's the time where ATP will enter the process. ATP here will give its energy or its phosphate to G3P. So we have 2 phosphate groups. And since we have 15 carbons that is being contained or carried by the G3P, and the ribulose needs to have at least five carbon. So we should divide 15 into 3. So we have a 3 groups of that contains 5 each 5 of carbon. So because of the phosphate that is being given by the ATP, it will look like this. It carries 2 phosphate and 5 carbons. So since it is stated there that a glucose should contain at least 2 G3P in order it to turn itself into sugar, we should at least repeat the process at least once, so that the 1 G3P here will be added with another 1 G3P and it will be the reason why Calvin cycle will give off or will have the product of sugar. So that's all in the process of Calvin cycle.